Welcome to our lecture on data retrieval systems in computational biology. Imagine walking into a colossal library that contains billions of books, but there is no librarian, no catalog, and no labels on the shelves. If you needed to find a single page about a specific human gene, it would take you a lifetime to locate it. Today, we are going to learn about hyper-intelligent digital librarians that solve this exact problem for biologists. We will explore what these systems are, how they work behind the scenes, and why they are the absolute foundation of modern genetic research. Let us dive in. To understand why we need these systems, let us first look at the scale of the problem. In the world of computational biology, we generate terabytes of data every single day. All of this incredible information is stored in massive databases, like GenBank or the Protein Data Bank. But having data is not the same as being able to use it. Without a proper system, finding a specific gene sequence is exactly like wandering through that massive, unorganized library we just talked about. You would be completely lost in an ocean of information. This brings us to the solution: data retrieval systems. Think of these as the ultimate hyper-intelligent librarians for biological data. Formally, a data retrieval system in bioinformatics is an integrated software interface. It allows users to query, navigate, and extract information from multiple interconnected biological databases, all at the same time. It is essentially a highly specialized search engine, much like Google, but specifically designed to understand biological terms and fetch exactly what you need in mere milliseconds. Now that we know what these systems are, let us look at the major platforms you will actually use. These systems work by parsing your queries using specific search fields, such as accession numbers, organism names, or gene symbols. Two of the most prominent examples in bioinformatics are Entrez, which was developed by the National Center for Biotechnology Information, and the Sequence Retrieval System, traditionally used by the European Bioinformatics Institute. These platforms are the industry-standard tools that researchers around the world rely on every single day. So why are platforms like Entrez so critical? Because they act as the fundamental bridge between raw biological data and actual scientific discovery. Before these systems existed, researchers were forced to manually download massive text files, known as flat files. They then had to write complex computer programs just to find a single DNA sequence hidden inside those files. Data retrieval systems eliminate all of that manual labor, allowing biologists to focus on biology rather than computer programming. Beyond just saving time, these systems offer something even more powerful: cross-referencing. Instead of forcing a researcher to visit 10 different websites to find a gene sequence, its protein structure, and the scientific papers written about it, these systems link all this data together using unique identifiers. For example, when a geneticist is studying a mutation causing a rare disease, the system automatically provides links from the gene to the three-dimensional structure of the protein it creates and directly to the medical literature discussing it. This interconnected network saves countless hours of manual research. This incredible speed and cross-referencing ability leads us to an important question. How do these systems filter millions of records so quickly? The answer lies in the underlying mathematics of search queries, which is governed by Boolean set theory. When you type a search into Entrez or the sequence retrieval system, the computer does not just read your words. It translates your words into mathematical sets. It then uses logical operators to find the intersection, union, or difference of these sets to pinpoint your exact data. Let us look at the mathematical form of this concept. The fundamental formula for a highly specific search query can be represented using set notation. Here, Q represents the final set of retrieved documents that the system will display to you. The variables A and B represent the sets of all records containing your first and second search terms. The intersection symbol represents the logical AND operator, meaning the system will only keep records that exist in both set A and set B. Finally, the backslash represents the logical NOT operator, which instructs the system to subtract or exclude any records that belong to set C. By increasing the number of AND conditions, the size of your final result set decreases, making your search much more specific. Now that we have our formula, let us see this with a concrete example. Imagine a biology student needs to find the DNA sequences for the BRCA1 gene, which is associated with breast cancer. But they only want the sequences found in humans. If they just search the gene name, they might get 100,000 records, including mice and dogs. So, we define our first set as all BRCA1 records, and our second set as all Homo sapiens records, which might be 5 million records. By applying the intersection operator, or Boolean AND, the system scans for records possessing both tags simultaneously. The final answer drastically reduces the search space to just 2,500 highly relevant human-specific records. Let us look at a slightly more complex real-world problem using the full formula. A researcher wants information on the p53 tumor suppressor protein in mice, but they specifically want to study the healthy wild-type protein, so they must exclude mutants. First, the system calculates the intersection of the p53 set and the mus musculus set, gathering all mouse-related records. Let us say this yields 10,000 records. Then, the system identifies that 4,000 of those mention the word mutant. By applying the difference operator, or Boolean NOT, the system actively removes those 4,000 records. The final answer leaves the researcher with exactly 6,000 highly relevant records, completely free of unwanted noise. To really cement this idea, let us visualize the mathematics we just applied. What you are looking at is a Boolean logic Venn diagram representing our search queries. The 3 circles represent our massive datasets. Notice the bright blue region where set A and set B overlap. This represents our intersection, or the Boolean AND operator. This is where the system found records that are both BRCA1 and Homo sapiens. Now, look at set C at the bottom, representing the mutant records. The large red X demonstrates the difference operator, or Boolean NOT, actively carving out and discarding that unwanted data from our results. This diagram perfectly illustrates how we surgically extract precise information from an ocean of biological data. Having seen the math and the visualization, we can now fully appreciate the impact of these systems. Data retrieval systems transform massive, unorganized repositories of biological data into highly searchable, interconnected networks of knowledge. They empower scientists to surgically extract precise information rather than drowning in flat files. Because all downstream analysis depends on starting with the correct data, mastering how to query these systems using Boolean logic is the very first and arguably the most crucial step in any bioinformatics workflow. Let us summarize the key takeaways from our lecture. First, we learned that data retrieval systems act as hyper-intelligent librarians, making sense of the terabytes of biological data generated daily. Second, platforms like Entrez and the sequence retrieval system do not just find data. They cross-reference genes, proteins, and medical literature into a unified network. Third, we discovered that the incredible speed of these searches relies on Boolean set theory. Finally, by mastering logical operators like and not, researchers can filter millions of records to isolate the exact data they need. You now understand the foundational tools of computational biology. Great job!